Okay, so I would like to thank the organizers for inviting me to participate today, and I'm excited to talk to you about our project on subtyping circulating tumor cells and breast cancer. So basically, we're interested in capturing a heterogeneous subset of circulating tumor cells, and then we have chosen to use a novel technology that allows us to isolate single circulating tumor cells that are appropriate for downstream analysis. So I am a fellow, a clinical chemistry fellow at the University of Louisville in the Department of Pathology, and my mentors are Dr. Mark Linder and Dr. Roland Valdez. So this project was done in conjunction with them. So unfortunately, I have no disclosures to report. And I'd like to also remind everyone that if you have any questions, you can go ahead and type them in as they come to mind, and then I'll answer them at the end of the presentation. So just to give everyone a little bit of an outline about where I'm headed with my talk today. First, I'll give a background on circulating tumor cells, including the FDA-cleared method to enumerate them, as well as touch on a few other technologies that have been developed to characterize CTCs. I'll talk a little bit about some of the clinical trials that are currently going on, and then I'll go into the three main limitations as we see the field of circulating tumor cells and what we're doing to advance those limitations. So what is a circulating tumor cell? This is a schematic of a blood vessel. So here you see the epithelium, the basement membrane, and then the layer of endothelial cells and the blood vessel. So in part B of this schematic, we see a primary tumor. And so circulating tumor cells are thought to be cells that have been shed from that primary tumor and have intravasated into the systemic circulation. Those cells then have the potential to form metastatic lesions in the patient. So these cells are very dangerous to the patient, more so than what's in the primary tumor, because most patients, the mortality comes from metastatic cancer and not the primary tumor. These cells are really poorly defined. I'll go into what the current definition is in a little bit, but we really feel that we don't have the best definition of what a circulating tumor cell is in order to capture a heterogeneous set of CTCs. These cells are very difficult to study because they're in such low abundance. So in 1 ml of whole blood, there may be 10 million white blood cells and 5 billion red blood cells, and a metastatic breast cancer patient may have on the order of 100 circulating tumor cells. So they're very difficult to study because of their extreme low abundance. And we're interested in studying these because they hold such promise of prognostic value to the patient. So what is a circulating tumor cell? The current definition, most people agree, is the definition that's given by CellSearch, which is the only FDA-cleared method for enumerating CTCs. And they define a circulating tumor cell as a large nucleated cell, one that is CD45 negative. So CD45 is a marker for leukocytes, so it is not a hematopoietic cell. These cells are epithelial cell adhesion molecule positive, or EpCAM positive, which is a marker of epithelial cells. The cells must also stain positive for cytokeratin 8, 18, or 19, which is an intracellular marker of epithelial cells. This definition really describes a specific type of epithelial CTC, and we believe that this strict definition may not allow all what really are circulating tumor cells to be detected. So we're interested in potentially expanding this definition. So how does the FDA-cleared method work as an independent predictor of outcome. So this test uses 7.5 mils of whole blood from the breast cancer patient. And in that 7.5 mils of blood, if the patient has 5 or greater circulating tumor cells, they're associated with a worse prognosis, or specifically less than 11 months of overall survival. But if they have fewer than 5 CTCs, they're associated with better prognosis, specifically greater than 18 months of overall survival. And this test has been validated over and over again, and these values are very consistent. But what I'd like to point out is that this test really only provides enumeration of circulating tumor cells. There's no more characteristics about the individual CTCs that are given other than how many epithelial type CTCs a patient has. And so we're interested in providing more specific characterization of a patient's CTCs. So I'm just going to go through a couple of examples of what CTCs, how they're being studied in clinical trials. So there are many clinical trials currently going on looking at circulating tumor cells right now. I'm just going to list a few that I think are interesting. So one that has just finished is a study that's looking at the efficacy of lapatinib in patients that have a non-HER2 amplified primary tumor but have a positive HER2 circulating tumor cell. So this study showed a reduction in HER2 positive CTCs, so that's very promising. In this particular study, no clinical response was determined, but these types of studies are very important to look at treating the patient based on their circulating tumor cells instead of just the primary tumor. We think that that'll give a better therapeutic regimen for the patient. Another example, this one's currently ongoing, is looking at an endocrine therapy index. So in this study, they're examining markers of endocrine sensitivity and resistance, and they've come up with an algorithm to predict the endocrine therapy index, or ETI. And this is for patients that have ER-positive, HER2-negative metastatic breast cancer. And so the goal of this study is to provide the best endocrine therapy for an individual based on their circulating tumor cells. And the last example that I'll give of an ongoing clinical trial involving circulating tumor cells is one using the CTC Chip device that I'll explain a little bit more in a minute. So this clinical trial is looking at comparing the EGFR mutation on the CTC to the tumor itself. And so everyone believes that CTCs have great promise to be used as a surrogate for a tumor biopsy because it's less invasive to the patient and can be done more often. So what are capture devices that are currently being used for circulating tumor cells? So as I mentioned, the FDA-cleared method, the CellSearch, is the one that's most widely used and has been extensively validated. In this method, they perform a CD45 negative enrichment, so they deplete the hematopoietic cells. They use a ferrofluid containing EpCAM antibodies to enrich for circulating tumor cells. So they're capturing only those CTCs that have that EpCAM marker, so those CTCs that are epithelial in nature. They then identify that circulating tumor cell is in fact a CTC by staining for cytokeratin 8, 18, or 19. And this process is highly automated, and it's the most widely used method for clinical trials. The limitations of this method are, of course, that the cell must express the epithelial marker EpCAM, and the cells must be permeabilized to look at cytokeratin because it's an intracellular marker. And so because the cells must be permeabilized, that limits the type of downstream analysis or characterization that can be performed. So we're interested in using a method that I'll talk about in a little while that eliminates the need for the cell to be permeabilized so that we can characterize, perform downstream characterization. So this is one of the new devices that's garnered a lot of attention to detect circulating tumor cells. This method is, the first iteration of this was the CTC Chip, and I believe this was a collaboration of MassGen and Harvard, I think. And they use diluted whole blood on what they are now calling, the second version is the herringbone chip. And here's a schematic of this device. And this device has garnered a lot of attention and looks like one of the new promising methods to detect CTCs. So the herringbone chip applies passive mixing of blood cells through the generation of microvortices to significantly increase the number of interactions between target CTCs and the antibody-coated chip. And they've showed the ability to produce efficient capture of CTCs using this method. So I believe that we'll see more with this device. Another one, and these are just examples, there are many devices that are being developed to look at circulating tumor cells. This one's known as the MAG sweeper, I believe, from a group at Stanford. They use a robotic arm to collect cells that are labeled with EpCAM antibodies. This is nice because it's very highly automated. And again, a limitation with this method is that not all CTCs express EpCAM, so they would be missing any CTCs that may not be epithelial. And then lastly, this is an interesting method, ISET. It stands for isolation of epithelial tumors. So they use size to enrich for their tumors. So they use diluted whole blood, and they use a filtration device that takes 3 minutes. And they filter out all of the smaller cells and leave the larger CTCs behind to be able to characterize and enumerate those. So a limitation with this method is that not all CTCs have a larger diameter. There may be some that flow through the filtration device and are lost. But this is interesting because it captures them purely based on size and then positively identifies them after that it's enrichment based on size. So what are the current limitations in circulating tumor cell utility? So the first one that I'll address is that I've been talking about circulating tumor cell identification is currently very based on epithelial markers. The next one that I'll talk about is methods. Most methods don't allow you to capture a visually identified cell. So those methods that allow you to visually identify them, those cells usually must remain on the cartridge. So that's a negative aspect to most isolation methods currently. And then lastly, the FDA-cleared method only allows prognosis to be made on enumeration of epithelial CTCs. And we would like to provide patients more information about the nature of their CTCs and provide a prognosis on the characterization of that individual's CTCs. Why would a circulating tumor cell not express EpCAM? So typically on the left is what epithelial cells tend to look like they grow in tightly packed rows. They have very strict requirements for communication and growth, and these cells tend to be EpCAM positive. However, it's thought that a process known as epithelial to mesenchymal transition occurs in cancer. This occurs not only in cancer, but also early on in development and also in wound healing. So these cells transition from this epithelial type cell to a more mesenchymal cell that's more elongated and is more highly mobile. These cells express metalloproteinases that can chew through extracellular matrix, so it allows them to to move more freely and without the tight requirements that are seen in epithelial cells. And these cells tend to express less EpCAM, so it's either downregulated completely or there's very little expression of EpCAM in these cells. And so why is this important? We've been able to see that there is heterogeneity within CTCs, not only between patient to patient, but even within a patient from a single blood draw. So there is heterogeneity within CTCs. And then there's also the fact that in metastatic breast cancer patients, 40% of those patients have no detectable CTCs using the current methods that identify CTCs based on EpCAM. And so we're interested in capturing heterogeneous CTCs and being able to find those CTCs in this 40% of metastatic breast cancer patients that currently have no detectable CTCs. And so in order to do that, we've developed a cell line method to capture heterogeneous set of CTCs. And we've included a couple other positive markers for identification. So in addition to EpCAM, we've included CD44, which is a stem cell marker, and HER2, which is a marker of growth. So this is a flow cytometry plot. So this is our breast cancer cell line model, and we've intentionally chosen cell lines to represent differences in metastatic potential and different molecular subtypes. So on the y-axis, we'll be looking at EpCAM expression, on the x-axis, CD44 expression. So the first cell line that we've chosen as a CTC subtype is MCF7, which is a luminal subtype. They express high levels of EpCAM and very low levels of CD44. This is the least metastatic subtype that we've chosen to include in our model. The next is SKBR3, which is a HER2 subtype. It expresses HER2 and EpCAM. The next that has a sort of intermediate metastatic potential is a basal A subtype, and it expresses all three of the markers that we've chosen for positive identification. And the last is MDA231s, which is a basal B subtype. It expresses CD44 and is the most metastatic. These metastatic potentials are based on cell invasion assays and how aggressively the cells grow in culture. And I'd like to point out that because MDAs produce a low level of EpCAM, the majority of them would be missed by the current method that captures cells that express EpCAM. And these would potentially be the most dangerous types of cells to a patient because they're the most highly aggressive subtype that we've chosen. So in some preliminary experiments that we've done, we've looked at the separation efficiency or the specificity of these four lines using those three markers. Here's another flow plot that we've looked at. This one, the y-axis is CD44, and the x-axis is HER2. So with these four cell types, we've determined the separation efficiency using 100,000, 10,000, 1,000, and 100 cells in repeated assays. So we see a high separation efficiency for MCF7 and SKBR3, and then lower separation efficiency for HCC and MDAs. But overall, it's about 80% separation efficiency. And so to represent what we would see in a patient sample, we've done spiking experiments where we've spiked our 4-cell line model into whole blood. We then perform a Ficol gradient to obtain a Buffy coat, and then we negatively deplete our cells with a CD45 antibody that has a magnetic bead attached to it. And that allows us to deplete the unwanted hematopoietic cells, the leukocytes, and then we're left with an enriched tumor sample. The next thing we do is then to treat those cells with magnetic antibodies that are conjugated to CD44, EpCAM, and HER2, and pass them through the column again, but this time in the flow through, we have our unwanted cells and our desired cells remain attached at the site of the magnet. We then remove the sample from the magnetic apparatus and are left with a highly enriched population of desired cells. Looking at that method, we looked at the MDA cells, so the cells that are that express EpCAM the least, so they have very low expression of EpCAM. And we determined in spiking experiments where we spiked in 100,000, 1,000, or 10,000 and 1,000 cells into 1 ml of whole blood, we determined that the percent recovery was 85% using that method. So now that I've discussed how we are capturing a heterogeneous population of CTCs, now I'm going to discuss the limitation that currently most of the methods do not allow positive visual identification and then the ability to retrieve that cell from the cartridge. So to combat that limitation, we have chosen to use a novel technology known as the Deborah Ray. This is produced by Silicon Biosystems to allow us to collect single viable CTCs. So this method allows cell trapping in individual dielectrophoretic cages. So dielectrophoresis is the process by which a neutral particle, such as a cell, can experience a net force of movement when it's exposed to a non-uniform electric gradient. And here's a schematic of this individual dielectrophoretic cage and one cell that's housed in it. So we can control the movement of these cages, which in turn allows us to isolate these single cells. So using the Deborah Ray, we can actually watch the cell being sorted into the container of our choice. So on the very right-hand side is a bright field image of a cell that's trapped in this dielectrophoretic cage. And because dielectrophoresis is a gentle method on the cells, it allows cells that are viable, and these cells were positive that we have what we're looking for because we're able to visually identify them and watch them on the chip as they are sorted and provided to us. So here's a cartoon of this Deborah Ray cartridge. So we add the cells, so this would be an enriched population of cells, and we inject them onto the cartridge. They're then sorted into their individual dielectrophoretic cages, and we then visually identify which cells that we're interested in. So say I'm interested in this blue one and this green one. I then tell the instrument to sort those cells to the parking chamber, and I watch them as they are brought to the parking chamber and say I want to look at the cells individually. I have the cell sorted individually into their own tubes. And so, as far as I know, this is the only method that allows you to positively visually identify cells before they're sorted. These cells can be grown in culture following isolation. These cells are intact, whole, and viable, and are appropriate for downstream characterization following this sort on the Deborah Ray. So what have we done so far with the Deborah Ray? What does this allow us to do? So in this experiment, we used a mixture of our 4 cell lines, and then we sorted them. We loaded them onto the Deborah Ray and sorted them using the Deborah Ray. So in this schematic picture, you're looking at one cell that goes all the way across in different channels. So this cell we were able to see is EpCAM positive and expresses very little or no HER2 CD44. So the fuzzy background indicates that we had to zoom way in to see any signal. So these are essentially negative or have very little expression. And then the bright field shows the cell in its cage. So next we were able to identify a cell that was EpCAM and HER2 positive but CD44 negative. And again, you see the cell in its bright field cage on the right. The next cell we were able to identify was positive for all 3 markers. We were able to identify that it was in its own cage, so it was single and viable. And lastly, we found cells that were negative for both EpCAM and HER2, but positive for CD44. So what does this tell us? It tells us that we were able to sort from a mixture of cells and determine what subtype of CTC, if you will, and then also the metastatic potential. As I've shown you before, MCF7 has the least metastatic potential, and MDA231 has the most metastatic potential. So using this method, we'd really be able to give a better indication of the metastatic potential of that individual's own CTCs with this method. And so lastly, the limitation that I'll focus on is that currently prognosis is made based only on an epithelial subtype. And so we're interested in combating this limitation and providing more information to the patient about their CTCs. So in this experiment, we performed the enrichment that I described earlier. So we spike cells into whole blood. We do a -45 depletion and then a positive enrichment. We then can use either the Deborah Ray or a single cell sorter, a cytometer, to sort single cells and then interrogate them on a single cell level. So this experiment, we're looking at transcript expression. And so we've devised sort of a metastatic potential based on transcript expression. So we gave points to each transcript that we looked at and devised an arbitrary score between 1 and 10, 1 being the lowest and 10 being the highest. And this was based on what's been observed in the literature. So in this plot on the y-axis, we're looking at metastatic potential in those arbitrary units that I just showed you. And on the x-axis are different CTC subtypes, or the different cell line populations. And so looking at an average metastatic potential of traditionally isolated RNA and then converted to cDNA, looking at a million cells at a time, we see that MCF7 has the lowest metastatic potential, SKBR3 and HCC1954 have intermediate metastatic potential, and that MDA231 has the highest metastatic potential. And this confirms what's been reported in the literature about these cell lines. So here's the same graph that I just showed, looking at traditionally isolated RNA, or the average metastatic potential. But once we interrogate single cells that have been sorted in a single cell manner, so each one of these represents, on the y-axis, the percent of single cells. And then on the x-axis, we're looking at metastatic potential, going from lowest to highest, 10 being the lowest, 100 being the highest. And so when we look at single MCF7 cells, we see that most of them do have low metastatic potential, but within those cells, some of them do have a metastatic potential of 50 or 60. So that's just indicating that there is heterogeneity on the single cell level from what was once thought to be a homogeneous population of MCF7 cells. And because we sorted these cells based on their extracellular protein expression, it is a little bit surprising that there's such a difference in metastatic potential between these cells. But this is just confirming what's been reported in the literature, that single tumors are heterogeneous in expression, and a patient's own CTCs within a single blood draw are heterogeneous. And so using this model, we'll be able to identify the heterogeneity in each person and in each patient, and provide them metastatic potential for their circulating tumor cells. And so now that I've shown you the cell line with the least metastatic potential, now let's look at the cell line, or the subtype, with the most metastatic potential. So MDA231s are the most highly metastatic cells. So on the y-axis, we're looking at percent of single cells, and on the x-axis, metastatic potential. So the majority of these cells do have high metastatic potential, 90 or 100, but a few of the cells had lower, so 40 or 20 metastatic potential. So again, this is characterizing the heterogeneity within what was once thought to be a homogeneous population. So we're finding unique subtypes within a single cell line, and those subtypes likely possess different functional abilities, and it's really representative of the heterogeneous CTCs that we're likely to observe in a patient. And this really speaks to our ability to go from whole blood down to a single CTC and characterize that single CTC to provide the patient with the most individualized information about their own CTCs. So in summary, I've shown information indicating how we're interested in identifying heterogeneous CTCs, and we're doing that by including a few additional markers for positive identification, and identifying CDC subtypes based on metastatic potential. We have discussed how we are able to capture visually positively identified cells using the Deborah Ray, and then those cells can then be used for further downstream characterization to provide the most information to a patient about their CTCs. And then lastly, we're combating that the prognosis is currently made only on enumeration of epithelial cells, and we're looking to expand prognosis to include heterogeneous CTCs to provide the patient with metastatic potential of their single individual CTCs. This will provide the best therapeutic regimen for the patient. Eventually, we hope to get to the point where we can identify what tissue those CTCs are likely to metastasize to, so that potentially that person could take prophylactic treatment to prevent that. But we really believe that using this method will provide the most individual information to a patient about their own CTCs and what therapeutic regimen would be best for them. So of course you can obtain continuing education credits by going to the bioconferencelive.com website. And I thank you for your participation and your attendance. This is a schematic of our cardinal bird in Louisville and one of the buildings that I work in. So thank you so much for your attention, and I'll answer questions now. So one of the questions that has been posed isn't a challenge to ascertain statistically significant heterogeneity in 100 CTCs. I think that there will be, if there are 100 CTCs in a patient sample, I think that there will be easily statistically significant information about a patient's heterogeneity. I don't think that would pose too much of a challenge to prove that it was statistically significant. Let's see some of these other questions. There's a question about how many dielectrophoretic cages are on the chip. So the chip houses 80,000 cages, so our sample must be enriched enough to have less than 80,000 cells. CellSearch is used for breast cancer as well as for colon cancer and for prostate cancer. So this question was asking if CTCs detected by CellSearch can apply to other cancers, and it's really just those 3 epithelial cancers so far. But many other studies are looking at trying to identify CTCs in lung cancer and other cancers that aren't epithelial in nature. So this question asks, why are we discarding EpCAM low negative cells during the enrichment procedure? So we're not discarding those cells, we're keeping those cells as well. We're just expanding the number of positive markers that will be available to detect those CTCs. And this last question looks like they're asking about next generation sequencing used for analysis of CTCs. And yes, I think that will be routinely used in the near future. We're working on, there's a question here about sensitivity and accuracy of the Deborah Ray. So I showed that we have about 85% yield, but we're working on looking at accuracy over many studies for capturing CTCs. And it looks like that's about it for the questions. So I thank you so much for your attendance, and I wish you all success in your scientific endeavors. Thank you.